have Craig Venter, and he's going to talk with Dimitar Saselov about his new book, The Voyage of Sorcerer II, the expedition that unlocked the secrets of the ocean's microbiome. And that's 18 words in the title. I mention that only because I am not going to introduce Mr. Venter, but Dimitar is. But I'm going to introduce Dimitar because he's my friend and colleague. He's not my friend, okay? He's my colleague. Sometime in the future, he may be my friend. And he's a professor, he's the Phillips Professor of Astrophysics, which is somewhere up there on a hill. And he, on a hill, and he did his, I'm just introducing him because he's kind of interesting and he kind of connects with the author tonight. Are we doing okay? Yeah, okay. So here's the thing, that Dimitar was born in Bulgaria, apparently by the sea. And he did his PhD in physics in Sofia. And then he, of course, made the great choice to come to Canada, to the University of Toronto, to do another PhD in astronomy. So he has 2 PhDs. You see why I want him to be my friend. And also, apparently, and this is something I didn't know until a few minutes ago, even though I've known him for a long time, he discovered a planet in the Milky Way which is the furthest from the Earth. And I'm not sure whether it's still the furthest because things have moved a little bit.
It did move.
Just a tiny bit from there. And it's called, the weird thing is called Ogle TR, which makes me think of Ogletree. Ogle TR 56b. Does anybody recognize?
Yeah.
Is it a habitable planet? Yeah. Anyway, he's the director of the Origins of Life initiative at Harvard, so he's interested in the origins of life. And he also works on, I think, NASA's Kepler mission, unless that's not true.
Well, that finished, so now it's another NASA mission. But
okay.
Yeah.
Weird, because I just read a paper about it. It must have been an old paper. Anyway, he wrote a book, and get this. Here's the title. The Life of Super Earths. How the hunt for alien worlds and artificial cells will revolutionize life on our planet, which is 20 words. These are the 2 longest titles, the titles that have been in our book series. Sorry, I would like now to ask Dimitar to introduce our guest.
Thank you, Melissa. Thank you very much. This is a great program, but we really are very, very lucky to have our guest here tonight. Dr. Craig Venter, formerly a visiting professor here, known for the decoding or the mapping of the human genome many, many years ago. I see a lot of you here who were not even born when he did that. But yes, we know the DNA of the human genome.
It was longer than that one, but no.
Really? Well, we have to update our classes here. Anyway, so that's the most important achievement most people my age know Craig about and should know because it has really revolutionized our ability to understand and treat disease, to understand where we come from and how we connect to our past in many ways, and many, many other things. He did that as a scientist, initially in the standard way we scientists are in the department at the university, then at institute in Bethesda. But most importantly, he is the founder and CEO and president of the Craig Venter Institute, which is an independent research institute, nonprofit, where a lot of this work was done. And that is still an institute which does all kinds of things genomic and even more, including the research, the science that was done as a result of the Voyage of the Sorcerer II, which is the topic of this book. So he found it, and he still runs it. How many years? 20?
It started in 1992.
Yeah, a long time ago. On top of that, of course, you started a number of companies, Celera, Synthetic Genomics, Human Longevity, probably others, but these are the successful ones.
And I was fired from all three.
You fired all three?
Yes.
Oh, I didn't know about the Human Longevity.
Yes, it gives me great credibility with some groups.
Wow, that's more than Steve Jobs.
Yeah.
And so I think what Craig and I decided is to have a conversation in which you will learn a little bit more about what he has done and his life. So I don't need to go into many details, like list all of his medals and honors, of which he has many. Most importantly, the US Medal of Science.
Thanks, yeah.
By Barack Obama. How many years ago was that?
2008.
Yeah, 2008, yeah.
Everything's getting to be a long time ago.
Well, with the pandemic, everything is compressed. So let's have the conversation. The plan is, we're already 10 minutes into it. We'll converse for about half an hour, me and Craig. And then we'll give you about 20, 25 minutes to ask questions. And we'll continue the conversation with you being part of it. So think about this. So Craig loved the book. And I want us to start by presenting the book in the scientific sense. Obviously, it's a voyage of a boat. And as Melissa nicely said, I was born by the seaside. So when I was a kid, just so you know, this kind of voyages was one of my kind of big excitement and dreams. At the time, this was Kon-Tiki and Thor Heyerdahl and Bengt Danielson going to the South Seas. And the science there was anthropology, history and anthropology. It was not what you did or what Darwin did in his voyage. This is more like the voyage of the Beagle and Darwin. But I want for people for our audience, to get in the conversation from the very beginning, understand why this voyage made sense, why you did it. So going back to Bermuda in 2002, the bar in which you actually decided with the others on the team that this is the thing to do and how to do it.
The bars in Bermuda are great places for scientific discussion.
Sounds like it, yes. So could you kind of give us a primer on why was this a problem worth tackling? And I mean, that was on the coattails of you finishing the human genome. So the human genome was done. You had done other genomes before that. But then there was the whole ocean, the whole Earth. But you decided the ocean is the right way to do it, which makes sense. But let's explain the logic behind all of this. So there were.
Let's start back a little bit earlier. So in 1995 is when Hamsmith and I sequenced the first genome of any living species. It was his pet bacteria, Haemophilus influenzae, that he discovered restriction enzymes and that, which led to his Nobel Prize in 1978. And we had to develop new algorithms. So I had this idea of shotgun sequencing genomes, because the way it was being done, each one was turned into a major public works project, and even E. coli, which was supposed to be the first genome done, was going to be a 5-year project costing tens of millions of dollars. And they would break genomes down into pieces and finish each segment. So Sanger did that when he sequenced Phi-X174, a small bacteriophage. And when we published the first genome, I sent him a copy of the paper, and he sent me back a really nice note that he always thought that approach would work, but always colleagues wanted to own their own piece of DNA. But the idea with shotgun sequencing is take the entire genome and we blow it apart. With a small bacteria, there's like 25,000 fragments that we sequenced. We get 500 or so letters of genetic code for each one. And so the trick was developing an algorithm to reassemble the genome in the computer. And so our team did that at the earlier version of the Venter Institute called the TIGER, or the Institute for Genomic Research. And to our amazement, the algorithm worked incredibly well. Basically, it looked for overlaps in sequences. The analogy is like taking the Sunday New York Times or 50 copies of it, shredding it, and then trying to realign all the fragments to reassemble the newspaper.
And the computer does this.
And the computer does it by looking for identical overlaps. And it worked better than we thought. After we sequenced the first genome in history, we were flooded with money and requests to sequence, every pathogen and every organism. We were sent a clinical sample from Norway from a patient that was refractory to antibiotics.
I wanted to ask you about that Norway patient, because that was a aha moment for you, wasn't it? Can you tell us about this a bit?
So we sequenced what we thought was the genome from this patient's blood, but the algorithm assembled two separate genomes,
The
circular
doctors
genomes.
thought he had just one pathogen, and you discovered two.
Yeah. And it's the first time it became really clear to me how powerful the algorithm was. And not only that, but it was the first notion I had that each species, in fact, in our case, each human, has a unique mathematical solution for their genome because of the uniqueness. We then went on 5 years later to sequence the human genome. For that, we had to scale up to 25 million fragments. So obviously, the computational problem got substantially larger. But to our amazements and everybody else's, it worked. And we had to build the third largest civilian computer in the world to do it, which seems comical 24 years later, because it was 1.5 teraflops. And now you can get a desktop computer to do that for not much money. This one cost over 100 million. And filled a room larger than this with things, but it managed to assemble these 25 million fragments. So it became very clear how powerful the algorithms were.
So you basically had a team that managed the computer. I mean, these units have to be managed all the time because they burn out and so on. And you had a team who wrote the software because you have to deal with the software.
They actually developed a new version of the algorithm to deal with the increased complexity. Gene Myers was head of the algorithm team, and we spent earlier today with him, and I was speaking at a symposium at Brown University in a couple of days, sort of celebrating this. The team's getting back together after all this time. But, I had the idea, but I couldn't have written any lines of code if my life depended on it. But the concept is remarkably simple.
And probably it's good for the audience, especially younger people, to know that when you were doing this software, companies like Google and Facebook didn't exist.
Right.
So nobody was doing this kind of large data analysis except the astronomers, I guess.
And physicists were. DOE had some of the largest computers in the government. But knowing how powerful the algorithm was, so I left Celera with encouragement after sequencing the human genome and raising $1 billion cash. I was deemed redundant, and I wanted to go back to my institute anyway. And I took some time off and sailed to the Caribbean and lived there for 6 months and sort of recalibrated my life. But I've always been a sailor like you and thinking about the ocean and trips. And I was reading these papers, one in particular in PNAS, arguing how little diversity there was in the oceans.
So that's an important thing to mention.
Yeah,
that was the understanding at the time that the ocean was a desert.
Well, and I have to take everybody back. Microbiology before genome sequencing was a field that used microscopes and culturing. if a microbe couldn't be cultured, it was deemed not to exist. So in the Sargasso Sea, they numbered them one at a time as they discovered them. They were up to SAR 11, Sargasso Sea 11. And that's why people thought there was, it was considered an ocean desert. And I wanted to try shotgun sequencing the environment. I was sure the algorithm would work. I was basically the only one on the planet who thought it would. I wrote a grant and submitted it to the Department of Energy. Every reviewer turned it down with extreme prejudice. But Ari Petrinos, the head of biology, was one of the few that believed, he was a physicist like you, that believed in biology and big computing and big ideas. And so he funded the project. And we just took a barrel of seawater and small filters that were about this big around, and filtered out the viruses and the small organisms, and then just isolated all the DNA and sequenced it. And instead of finding 7 organisms, we found several thousand. And we stopped sequencing after roughly 1.5 million new genes just from this one barrel of seawater. So that was sort of the seminal study that turned all the thinking upside down.
And again, for the benefit of the audience, new genes, what you mean by that is you had a database before that already.
Well, there are public databases, GenBlanks.
And you would not find any gene that corresponds to this.
That's right.
Yeah. So these were potentially microbes or viruses which had functions or expressions that were not represented in anything else that we knew before that. Is that how?
So now we're up to sitting in the bar in Bermuda. I mean, it wasn't just one occurrence, but simple questions. And as we've talked earlier, science is composed of asking simple questions. The answers usually aren't so simple, but the questions are. And so the question was, if we sequence other sites in the ocean, would we just find the same organisms? Was it a giant homogeneous soup? Or would we find diversity? Nobody knew. But there were a couple of major expeditions early on. Darwin wasn't really on a scientific expedition. He was on a survey ship that naturalists would go on these and make observations. And what I call it observational science, which you can't get funded in this country to do observational science, maybe unless you're a psychiatrist or something. You observe people, but...
in astronomy, we call this fishing expeditions.
In biology, it's called
funded,
fishing, and you can't get funding for it. But the first scientific expedition at sea was the Challenger expedition in the 1870s. And the brilliant thinking at the time was there couldn't possibly be life below 1,800 feet in the ocean. And what the Challenger did, it set out with a very long line and a dredge that went to the bottom of the ocean every 200 miles, roughly around the equator. And every place they sampled, they found new life. Also, this was the time of the first transatlantic cables, and people wanted to know what the bottom of the sea really looked like. And so that changed the thinking about the diversity of life on the planet, but it was all macro life, things that, like Darwin, could see with his eyes and touch and feel and pick up. And so we decided to follow the Challenger expedition route, starting in Halifax, and sample every 200 miles. So you might ask, why every 200 miles? And sailors know the answer. It's roughly how far a decent-sized sailboat sails in a day. And instead of putting a dredge down, we took on 200, 400 liters of seawater and filtered it. And what we found, every 200 miles, things were 80% unique. There was so much diversity and so many microenvironments. I mean, there's tens of millions of them, which, if you've been to sea, which you have, or lived by the sea, you know it's always changing. There's winds, there's currents, different amounts of sun, rain, et cetera. So probably if we sampled every mile, we would find diversity. And like the first sample in Bermuda, most of the sequences were totally new and unique. And the data kept piling up, and we could assemble genomes of all these unknown organisms. We didn't know what the organisms looked like. We knew their genetic code. And one of the most unique findings was, instead of all these being unique punctate species like we're used to with microbes, they would, for example, if we took a reference genome, such as SAR11, we sequenced SAR11, and then we'd see all the sequences in the database that would line up against this reference sequence. And we found there were clouds of species, tens of thousands in some cases, but the sequence would vary as much as 50%. Now, we're 10% identical to all mammals, so 50% is a lot of sequence divergence, but very similar genes across the entire genome. And these were all photosynthetic organisms. We get 50% of the oxygen that we breathe from these organisms. And what I reasoned is the two major causes of mutagenesis are UV light and oxygen.
Yeah.
And so these organisms are constantly being mutated and creating versions, new versions, which is essential for their survival. And it turns out it's essential for our survival. If they disappear, we disappear. And one of the most interesting findings, so if you go out in the river here, the sea, every milliliter of water will have 1 million bacteria and 10 million viruses. So there's 10 viruses to every bacterial cell. Yeah. And you may ask what these viruses do, and what they do is they control the population of their host, but they don't want to kill their host.
Right. I want to talk to you about viruses specifically. Yeah. It's a great topic, yeah.
So one of the most important findings we did with the viruses was found that they actually carry new copies of the key photosynthetic genes, and they update the software in their host cell. So they want to keep their host cell live and happy, mostly keeping the photosynthesis going. And so it raises philosophical questions of, who's in charge in biology? So, it makes you think the viruses are, because they're working on keeping us alive so we can grow more of them. But
I have questions about the viruses, and I'm sure you do too, because we are now very painfully aware about viruses. And
but very few affect humans. I mean,
of course.
Less than a fraction of a percent affect us.
So
most of them keep us alive.
Yeah, every living thing essentially has viruses that can affect it, or that are matching to it, bacteriophages, viruses for the bacteria and so on. And it is a whole universe there. And you say in the book, it's difficult to call them alive because obviously without the host,
they can't replicate.
But then it seems to me, again, from the origins of life perspective, like the astronomer's naive perspective, looking at the planet as a whole, is that you cannot have life of any form, cellular or whatever, which has genetic material, without the existence of a tail, and actually a very long tail of those little fragments, whatever their origin is, which then become the viruses. There is so much trial and error going on in the biosphere that it is clear to me that there are a lot of viruses that are duds, that are kind of plasmids that escaped or were from dead cells, and they just disappear. But the ones that work then suddenly become, viable populations.
Well, and part of their
is
infection.
That your view of it?
Yeah, they always
from a planetary point of view.
They always pick up DNA from their host cell, and it's called lateral gene transfer. They move it around like new copies of these photosynthetic genes, and that's how we keep getting tremendous diversity, as well as just renewing the genetic software, as I call it, of their host cells. And it's not clear at all what came first, but there's not a single niche on this planet that you can't find microbes in. And we were all sort of brought up thinking a human-centric form of life, but we have microbes that we found that live in pH 11 water, pH 1 water. If you put your finger in it, it will dissolve the skin. Ones that can take huge doses of radiation. They can actually be found in nuclear reactors. I angered the head of NASA early on when I said the outside of the space station was covered with human feces, which it was, because early on in the space station, they would just pump the waste outside the space station. And apparently it didn't go far, or hopefully it didn't, or, you're doing spacewalks, you couldn't run into interesting things. But they stopped doing that, and now they pack the human waste into stainless steel containers and shoot them back into the Earth's atmosphere. So I always tell people to be careful about wishing upon a shooting star, because you may get a very, very different outcome than you expected. So it's, but they're in every environment, everywhere, and I delight in telling astronomers and physicists, that we found more organisms in the ocean than there are stars and galaxies in the entire universe.
Now you know why I'm interviewing Craig. That's how Melissa figured it out. Chapter number 8. There are more microbes than stars, which is true. In fact, there are more microbes than stars and planets together.
Yes.
Which we know there are a lot of planets in the galaxy and in the galaxies as a whole. But let's go to that. Actually, that's an interesting point. So the result of your voyage and all the sampling you did is this mind-boggling diversity and number of new species. Mm-hmm. So now we know that the oceans, and we know the soil in the continents, and the bottom of the ocean, and below the surface of the ocean bottom, and in very deep mines. There are microbes everywhere.
We've sampled a mile deep in a coal bed methane mine in Colorado. But there the diversity is very different. There's diverse sets of microbes, but we don't find these clouds of organisms like we do in the ocean.
There is no
mixing. There's no oxygen and there's no UV light a mile deep in the earth. Okay. It's high temperatures. One of the organisms we found in the mine in Colorado, the closest relative we found in a volcano in Italy. So, and many people argue there's more subsurface microbes than there are soil microbes.
Right.
So we have this tremendous diversity.
What do you think? Do you think that's the case? There are more subsurface microbes as a biomass or as a diversity, or?
Well, it goes down till you get to temperatures over 130 degrees centigrade, and then you see sort of a disappearance of life, but that's pretty deep. That's a mile or two in general. So that's a pretty large volume. And the deep vents in the ocean, every place anybody has looked, they find microbes. And in 1996, we sequenced the first methanogen from a hot vent very deep in the ocean, and takes CO2 and converts it into methane, which doesn't help the planet very much.
But again, talking about the number, so the number that you found, and now is kind of the census of all microbes, is 10 to the 30, which is 100 million times more than all the stars that we know in the visible universe, not just in our galaxy. And by the way, we actually know that stellar number pretty well. So the question to you, Craig, is do we know the 10 to the 30 number pretty well? Is it just the tip of the iceberg? What do you think? Is there still
we could be off by a few orders of magnitude with a number that large. We're taking the volume of the ocean and the density of microbes that are found and the density on soil. So it's probably within an order of magnitude or 2 or 3. But the weight of all the microbes on the planet exceeds, I forget the number, by 6 or 7 orders of magnitude, the weight of all the plants and animals.
Yeah.
So, and each one of you have about 3 or 4 pounds of microbes in your body. We use the same technique for sequencing the first human microbiome and found the same type of diversity. So we are a host for a lot of microbiota as well. We live in a microbe world, and as Dimitar knows, I've argued when we can sample on Mars and all the planets he's discovered, I'm certain we will find microbial life. And it'll be similar to life here, because in fact we exchange roughly 100 kilograms of material between Earth and Mars a year. So you can't take a shovel of Earth here without having Martian soil in it. And so the oceans and the apparent oceans on Mars, there has to be life in them. My view is any place we find the same chemical environments, we will find life.
But it's, from the astronomer's perspective, to me, that's one of the conceptually important things that I learned from the book, of course, from the papers and the work of that, is that planets on which the chemistry leads to life actually are completely, very quickly and completely taken over by those small unicellular species in the water, through the air. As you point out in many of your analysis afterwards, there is a lot of mixing and exchange. So they don't remain localized for very long. They expand very quickly. And so from a point of view of the origin of life, the initial stages, so to say, when all this eventually took off, in my sense, and I want to see what you think, is that it took off very quickly. It was kind of an exponential. Once it took hold, then the whole planet got infected, so to say.
Yes.
Is that a good way to think of it?
I wanted to create a bumper sticker that says, life happens. and yes, I think it happened very early on, and that's why we have this tremendous diversity that lives in almost any environment you can imagine.
So if you were to say, well, okay, I have in mind another similar project to do on this planet, where would you look? Where would be a set of niches or a niche which hasn't been explored enough? Just like 2002, you started with the Sargasso Sea.
Well, the deeper ocean has not been, it's been sampled, but it hasn't been fully explored. It's fairly easy to send pumps down to collect the first 100 meters or so, but then you start to need much more specialized equipment to get deeper and pump the water up. And some organisms, they're called barophiles, that live under this tremendous pressure at the bottom of the ocean or in the deep ocean. But when you bring them to the surface, they explode. So you can't just bring a sample up and have them stay intact. So we need a way to sample, and we're trying to develop autonomous vehicles that could do the filtration deep and then bring the filter up so we could isolate the DNA.
So you do the filtration at depth?
Yes.
Where the creatures are still in their own pressure and environment. Yeah. And
if they burst on the filters and you get the DNA, which is what we want. Yeah. So it's
So have you done any of that already? And
only with submersibles like Alvin and others, where you can bring it in and keep it under pressure.
So what is the general result of this kind of studies? Because of the UV, the sunlight, and other quickly changing surface conditions, you find more diversity near the surface than deep in, like in the mines?
We find in the first 100 meters is where we find these clouds of organisms from all the mutagenesis, I think, from UV light and from oxygen. As you go deeper, you find species diversity, if species even means anything anymore, but you don't find the same clouds. So Like in the deep mines, we'd find isolated organisms, but you wouldn't find the genetic variation where lots of copies of them. It's very stable under the pressure, the temperature, the lack of oxygen, the lack of other mutagens. So the deep earth probably gives us more hints about early life than what we find in the ocean that's constantly being mutated.
Yeah, yeah. Well, it's probably interesting for many in the audience. How do you see kind of us humans with our own microbiome, which we carry everywhere we go, existing in that environment where most of the microbes are benign and there is a whole cloud of viruses surrounding that. But you still, especially nowadays, a lot of people move continents, they travel nowadays. Is that introducing, apart from the obvious things, epidemics?
I mean, certainly pathogens move quickly like that with pandemics, as we've seen. But think of what's gone on for centuries with ships. Ships, when they get into port, they unload their goods, they take on millions of gallons of water to maintain the ballast. And then they go, across the ocean to another port, pump out that ballast water, and take on a new load of things. So we've been, contaminated and mixing things and changing the diversity on a constant basis ever since there was shipping and movement of things, at a gross level compared to what any human can carry with them. But what humans carry with them is very important. So we sequence from all the filters in the space station, and I would not want to go there. It's truly a microbial zoo. I mean, just you think of all the different countries that send astronauts up there and visitors, and they're all carrying their microbiome with them, all the bacteria on their skin, their mouth, they're all different environments. And so we've already contaminated the moon, we've contaminated Mars, because you can't truly sterilize any of these things we send into space. I argued with NASA that we should sterilize any astronauts and give them all the same synthetic microbiome so that they're not taking all this biota up there, at least until it's established what the unique life is on those planets. Once it's contaminated, it becomes very, very difficult to answer that. And we've had that problem. We sampled extensively in Antarctica, and we sampled from this frozen lake. And you have to make sure you're not introducing contamination just as you go into it. The Russians badly contaminated this lake.
Yeah, Vostok.
Vostok. And, but we sampled from one, and the density of microbes was 10 times that of the ocean. So this freshwater lake underneath Antarctica, just teeming with life. But we have to think of, sampling the supposedly deep oceans on Mars, or I love the early planets that he discovered with his colleagues. I love the ocean planets. We're an ocean planet, but he has some that are just water worlds.
Yes, water worlds.
And they absolutely have to have life. But my previous book was called Life at the Speed of Light, and my institute has a patent on a biological teleporter. And what I've argued, we can send a small sequencer. Now they have ones that fit in the palm of your hand. We send it to Mars or other places and sequence the microbes there versus trying to send up big rocket ships to bring samples back, which is crazy. We can just send them back, as I said, at the speed of light.
Yes.
And we can reconstitute the life here by remaking the organisms of the species, as we've done. So, I think if we can get portable sequencers to some of these planets,
yes.
I guess not in our lifetimes. It would be too slow to get the information back, even at the speed of light. But eventually life will be discovered everywhere. But
speaking of that previous book, this is your third book.
yes.
And each book is about the project, more or less, the human genome, then digital life, and now the diversity of the life on Earth, the microbial and generally life on Earth. So I know parents hate to be asked that question, who is their favorite child, but do you have a favorite project out of these three?
So probably the truly favorite one is when we made the first synthetic cell and the first minimal cell, which now has become a very critical research tool. There's now at least 50 major labs around the world using our minimal cell. And I've argued with that cell, we should be able to recapitulate all of evolution. If we get really good, we should be able to turn that little bacterial cell into a human or an animal someday. But thus far, various scientific teams have added genes back to the cell. The most interesting set is they found the genes for cellular mobility, and the cell started crawling around the plate. So by adding back things to a minimal cell, but the problem with the minimal cell is roughly a quarter of the genes are of unknown function. All we know is if you take that gene out, you can't get life. So, the scientific community and our population gets, reaches plateaus of extreme arrogance that we actually know the answers to things, and that, we're accumulating so much knowledge, we must have these answers. And I think what the data in the expedition showed, what the minimal cell shows, is how little of biology we understand. A quarter of genes essential for life, we have no idea what they do. With AI and some of the new algorithms predicting protein structures, we've solved, working with teams around the world, roughly half of them. But it's still a long way to go, even for that one cell. We have a large number of unknown genes in our own bodies that we try not to do the same experiment of removing those to see if you still live, but some of those happen naturally. And so it's, we have a very long way to go, and I think that's what this study helped prove, is how little we understand the environment. And also, I have a bias in science, like the discoveries we made. Most major discoveries are overcoming bullshit ideas from the past. Yes. With the human genome, only roughly 2% codes for proteins. So some genius has called the rest of DNA junk DNA. It's such an arrogant idea. And before we sequenced the genome, people thought there would be hundreds of thousands of human genes, one gene for every human trait and function. And people were very upset when we found, that some plants have more than the 20,000 human genes that we have. So just overcoming the ignorance of the past, like the Sargasso Sea was not a desert, is what leads to new discoveries. But the good thing about science versus politics, it's self-correcting.
Well, let's hope that...
Eventually.
Eventually. So let's hear the questions from the audience. I see there are a lot of them already.
And we should also talk about some of the adventures, including an adventure Dimitar and I had in Bulgaria with the Bulgarian mob. So,
well, I've planted some questions, so I'm sure they'll ask about that. Okay, so where are the microphones?
Right there.
Okay.
I was wondering what your thoughts on the deep sea mining expeditions are, and how that will affect the ocean microbiome in the water column and in the deep sea.
There's so many groups that want to do environmental engineering as ways to change our environment that we're doing tremendous damage. We found hypoxic zones with no oxygen, the largest one in the ocean's the size of Africa. And they're getting bigger from all the pollution and stuff. One of the problems with deep sea mining is there's huge methane deposits. And if we start knocking some of those loose and all that methane goes into the environment, we're going to go downhill very rapidly because methane is, 20 times worse than CO2. So usually we do damage before people find the right way to do things. We have plenty of places to do mining without risking destroying the ocean. People want to do acidification and things to...
Like the concept in human longevity is wait to fail.
Yes.
If you wait to fail, you have to treat a disease. If you do it in advance,
Yes. Yeah.
Another question there.
You mentioned bacteria and virus. What about fungus? Did you find much in the ocean or in the deep? I mean, is it as varied and as widespread in ecosystems?
Certainly less in the ocean, but there's lots. We were going for the simplest forms of life, the viruses and the single cell organisms. There are larger single-cell eukaryotic cells. Some of the most beautiful are all the diatoms that make these beautiful structures. So the eukaryotic genomes are much bigger genomes and much more complex. So the same amount of DNA sequencing yields far less. For example, when we do the microbiome, it's contaminated with human DNA. And you have to filter out the human DNA versus that. But there's tremendous eukaryotic diversity, not just the non-nucleus containing cells. More fungi in soil or marsh environments than the open ocean.
Yeah. Okay, there are questions down this side. Let's start here and then down.
Hi, thank you. When you're doing something like that shotgun approach to multiple organisms that you haven't encountered before, how do you know which genes, once you put the fragments together, went with which organism when it's all new?
Because it's a good question, because the sequences are so unique. One of the key tenets with the algorithm we use for assembling the human genome is the algorithm only put pieces together with, out of all the potential matches, there was only a single correct match. So if there's mismatches, we wouldn't put the pieces together. If you have 50 base pairs of sequence, that's what you need in the human genome to have something virtually guaranteed to be a unique stretch of sequence. In microorganisms, it's much less. In fact, because of all the mutagenesis and diversity, that we only need to do overlaps of 20 bases or so to have exact matches.
50 bases is about what you see here in the model, by the way.
But if you have multiple organisms that you're collecting the fragments from at the same time, even once you know that you have a new gene here, a new gene here, a new gene here, how do you know that gene A, B, and C went with organism 1 and gene G, J, and X went with organism 2?
So the overlaps give you what are called contigs that give you order and orientation of the genomes. In the case where you have enough fragments, we completely assemble the entire chromosome and you get all those answers.
Yes, thank you. I
One
was
thing
thinking
is
individual
you have more than
genes as
one
opposed to
microbe of
entire
a given
genome.
type in your sample.
Yes.
You don't have one of each. You have multiple of each.
Yeah.
Thank you very much.
Yeah.
So there was a question here. Yes, down there.
I would be curious, if I understand correctly, what you do is that you take these organisms back and you sequence them, which means blast them away. And would there be ever value of keeping the organisms? And I understand that you can actually revive them if you have the full sequence, but is there anything lost in that process if we don't keep the organism but only reconstruct the genome? And if I may have a second question which is related, can you maybe speak a minute about what could we do with that beyond the fact of the intellectual excitement of, hey, we have diversity, which is really fascinating. I'm there with you, but what else, what could we do with this knowledge?
So the first is literally impossible to do. We're talking about hundreds of millions of unique cell types. And to try and most of these would not grow in culture. We don't know the conditions. Quite often they're symbionts. They need other microbes that produce nutrients to keep them alive. From the genetic code, we can recapitulate the organism like we did with synthetic cell, just making it and booting it up. Or you can use the genetic probes to go out in the environment and find that exact organism if you wanted to grow it. Which is important for your second question. One of the things is these organisms in the oceans have, they exist with chemical warfare. They're always trying to attack their neighbors and survive. That's where a lot of antibiotics come from organisms trying to kill off other microbes. They have pathways that can do more complex chemistry than our best chemists can even imagine. So to me, the future of chemistry is taking these metabolic pathways out of the databases, putting them in synthetic cells to make new libraries of these chemical compounds that chemists couldn't possibly make. And I think that's going to be the basis of potentially a new chemical revolution and even a new industrial revolution. And what's changing, it took us 10 years to make the first synthetic cell, because writing the genetic code is much more complicated than reading it. But now there's new technology coming that makes it 10,000 times faster to write the genetic code. So because of all the unknown genes, we had to do a lot of trial and error until we got living cells. And now we'll be able to do maybe 1,000 chromosomes at a time, and you just screen for life. So you could screen for chemical production or others. So it's an important question about the future use of all of this. And I think it has tremendous potential.
A question here and then there, and then there.
Talking about sequencing to identify microbial lives, do you know if there's a means to distinguish Earth-originated microbial life from extraterrestrial life, hypothetically landed on Earth by way of panspermia? I know you would probably argue that they must be DNA-based, but they don't have to be DNA-based or even
Yes, they do.
The replication mechanism is not restricted to nucleic acid. If they are not nucleic acid-based life,
theoretically, but it falls apart pretty quickly. We find the same chemicals everywhere in the universe, and but the only way we can distinguish extraterrestrial life is discovering some extraterrestrial life first, and seeing if it's unique to that extraterrestrial environment, or because Earth has already been contaminated for billions of years from particles from Mars and other places. Panspermia is very likely, whether it came from Earth to Mars or Mars to Earth or other planets. It's been going on for a very long time. So we can't distinguish from looking at the genetic code. People who looked at the first methanogen we sequenced wouldn't have recognized it as a life form similar to anything they know. That's what Carl Woese discovered, was the archaea was this third branch of life that's halfway between prokaryotes and eukaryotes, and they look very different. The same is true for this bullshit article that claimed the COVID virus could not have been a synthetically made virus. In 1 week, our team made 50 variants of it for pharmaceutical companies to test new vaccines on, and there's no way to distinguish looking at a synthetic chromosome now from a naturally occurring one. There's no, markers of, that was 50-year-old thinking.
Some physicists are working on isotopic markers. So you may be able to distinguish by the very fine structure of the fractionation of isotopes.
Yeah.
In your lab, the isotope fractionation is different from another lab's. But that's a work in progress.
Yeah. And we looked at that with gene expression, with the anthrax genome, trying to work out where it came from. So, we need to sample one of his exoplanets before we can answer your question.
There's no fundamental reason to assume that they're DNA-based or RNA-based.
That's a pretty good assumption. I'll make a side wager, but we won't live long enough to find out, probably. So.
Yes.
So is the first synthetic cell evolving? Are new genes being generated in that cell?
So it's a good question. One lab did a study to see as they kept growing, did it mutate and change? And the answer is yes. All DNA-based organisms are constantly mutating and changing, including yours. Whether it's evolving, it's not clear. It so far hasn't developed any apparent new functions in these things, but the genetic code has constantly undergone some degree of mutation, sort of at expected rates.
Well, that's an interesting kind of point, which connects to the viruses. I mean, it reminds me of what Carl Wills wrote, about 15 years ago, about before there were restriction enzymes in the early evolution of life, the very beginning. Before the invention of restriction enzymes, it was very similar, like cells were exchanging packets of genomic information very freely because, they didn't even need viruses probably, but viruses do it today.
But so that happens all the time. So we see some organisms, and cholera is a good example. There was a big debate before genomics in the cholera community whether cholera had 1 or 2 chromosomes. As soon as we sequenced the algorithm, assembled 2 separate chromosomes. Carl Woese argued, because we had this committee advising which genomes we should do first, he said, don't waste time sequencing cholera, it's going to look like E. coli. But it's clearly phenotypically very different and causes diseases and lives in the ocean. There was 1 chromosome that looked very much like E. coli, and the other chromosome was very different. And we see this all the time, cells with multiple chromosomes that are different. They even have different 16S RNA that look like we're just fusion of 2 different cell types, which is how we got eukaryotes, plant life from one of these single-cell photosynthetic organisms going into a eukaryotic cell, and chloroplast are basically mini microbial cells. But to get you more interested in the book from just the science, it was also a tremendous adventure. The political ramifications were as important as the science. We got arrested twice. We got worked over by the mob in Bulgaria that Dimitar went to the high levels of government to save us. And he should tell this story because I've learned things from him after we left that people still talk about it. So.
In my next book. But yeah, that's fantastic. It's such a voyage. Do you miss Sorcerer? I understand it's no longer a research vessel.
Yeah, it sailed up to our house in Maine last summer, and we had a reunion with the captain and crew, and it was nice. But after 65,000 miles at sea, we have a vintage sailboat that we do racing, but I've crossed enough oceans now in my life. I'm much more choosy how I spend my time. So it was a fantastic adventure, and it's truly it's half science, half adventure story, and Darwin could freely go where he wanted and sample what he wanted. In fact, fortunately for Darwin, there was not a PETA organization back then. Some of the experiments he did in the Galapagos, he wanted to see if the iguanas could really breathe underwater. So he would tie rocks to them and throw them in the water to see if they would live on the bottom. He would swing them around by the tail to see how far he could throw them. If we did experiments like that, we would certainly not be able to publish the data in Science. There's the law of the sea that countries own all the genetic repertoire out to 100 miles off the coast. And so we had to work through the US State Department to get permits to take samples, work with their militaries at times. But sometimes we ran into geopolitics that had nothing to do with science. For example, the French government in Paris was disputing with the French Polynesia government in Tahiti as to who had rights to give us permits to take samples there. So we got arrested by the French, and the French military threatened to sink us if we left the harbor. We had to get the US Navy to intervene, the way you intervened with the police in Bulgaria, or the government. I guess we couldn't trust the police either there. But we had on the trip through Turkey and Greece and then into the Black Sea, we had 2 Navy SEALs for security. The only time they were afraid was in Bulgaria, with the Bulgarian mob in his hometown, so we had all kinds of levels of excitement just trying to take microbial samples around the world.
Has it become easier or more difficult?
It's getting more complex because we showed the value, but it started with the first sample in Bermuda. We were accused of stealing the genetic wealth of Bermuda. And we're putting all the data in the public domain. And some countries even complained about that because they said we were destroying their ability to patent it. Australia insisted we put a marker on every piece of genetic code saying property of the Australian government. And if you wanted to do something with that piece of DNA, you had to contact the Australian government. So the geopolitics of science is as intriguing as the science itself.
So I really like this. Sorry, I know I like a lot of things, but I really like this a lot. And I don't want to stop it, but I also do want to stop it. But I would like you to stay with us, as they say on NPR, and come in, maybe get a book and eat a cheese cube and talk further. Are these stories about the police in your book?
Yes.
Yes. Okay. So here's a good reason.
Half the book is adventure stories.
Okay. I'm just thinking, I don't know if you're thinking this, but I'm thinking maybe I could just retire and join the next adventure.
We've had lots of volunteers.
Have you? Oh, sorry. Okay, I know. I'm kind of old. Yeah. Okay. So I want to really, really thank Dr. Craig Venter and my colleague and possibly friend in the future, Dimitar Saselov, for doing this. Please stay with us and come across to the Cabot Library and indulge in books and cheese cubes.
Thank you. Thank you.